today's panel presentation, Harnessing Novel CRISPR Systems for Transcriptome Engineering. This presentation is part of the 4th annual CRISPR virtual event. I am Antonina Salcido of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots. Now let's get started. Before we begin, I would like to remind everyone that this event is interactive. We encourage you first to participate by communicating with other attendees using our new live chat feature during the presentation. You can find the live chat located at the right of your screen. You can also participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Help Desk button located at the bottom of your screen within the navigation bar or from the lobby. Finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits link located in the Abstract window below the presentation window and follow the process to obtain your credits. I now present today's speakers, Dr. Omar Abudayyeh and Dr. Jonathan Gutenberg, both fellows at McGovern Institute for Brain Research at MIT. For a complete biography on our speakers, please visit the Biography tab at the left of your screen. Dr. Omar Abudayyeh, you may now begin your presentation.
So, thanks for having Jonathan and myself today to speak about some of the research from our lab at MIT. I'm going to give the first half of our talk on harnessing novel CRISPR systems for sort of in vitro applications such as diagnostics for healthcare. And then I'll pass it on to Jonathan, who will go into a lot of our new CRISPR enzymes and their applications for RNA-based therapeutics, including a paper we just published in Nature on Cas711, the newest sort of RNA-targeting effector in the toolbox. So I'll go ahead and jump in. So, a lot of our motivation for our work comes from the greater understanding we have of human biology. So, of course, as more genomes are sequenced, we're understanding that there are more and more genetic differences that can be harmful or beneficial in both, healthcare and beyond. So, in agriculture, climate change, energy, and even more. So, the big need, as we understand that genetic variants can have effects on almost every aspect of our lives, is that we need tools to probe these variants, understand how they function, and then install the ones that we want to treat disease or for other functions. And so, to make a search function for both not just the human genome, but even for cells and engineering cells and beyond, we really search sort of the natural world. And so, we spent a long time mining prokaryotic genomes and bacterial genomes, sifting through metagenomes to really find new types of enzymes and tools we can leverage for biotechnology. And of course, there's a rich history for doing this. Of course, there's so many enzymes out there that have been used for molecular cloning, like restriction enzymes, fluorescent proteins, viruses for delivering genes to cells, and of course, gene editing and gene therapy. In the CRISPR world, we have spent a lot of time with Cas9 and more recently CPF1 and Cas12s that are useful for DNA editing, but they're really only a small slice of the whole sort of picture. And so, there's a huge diversity of other CRISPR enzymes. And so, we've spent the past many years sort of mining these CRISPR systems and using signatures of conserved genes that occur around these systems like Cas1, Cas2, or even CRISPR arrays to basically expand our understanding of CRISPR systems. And using these pipelines that basically look for seeds of signatures of CRISPR systems and then find new proteins around these seeds, we've identified many candidates of new systems. And so examples of these new systems, such as the type 5 Cas12s, the type 6 Cas13s, or CTC2 is shown here, and even more recently, the Cas711s, which fall in the type 3 world of CRISPR systems, which Jonathan will dive more into in his part of the talk. So basically, one of the big things we spent our time on are characterizing these RNA-targeting CRISPR systems. And so Cas13 is one of them, specifically Cas13A, which is kind of the first one that we found. And what's unique about Cas13 is that we found that it actually can target nucleic acid much like Cas9, but instead of targeting DNA, it targets RNA. And when it binds its RNA viral genome, it'll cleave, but it won't just make a precise cut like Cas9. It actually makes many cuts, eventually degrading the viral genome and actually cleaving even non-targeted nucleic acid, because it'll just turn on and cleave anything in the cell. This promiscuous activity is interesting, and how we first sort of uncovered it was looking in test tubes at the function of the enzyme. What we found is we could label these collateral RNAs, these green ones that are not targeted by the CRISPR RNA, with a fluorophore. And when we spike in a targeted RNA, as shown here in black, it activates the Cas13, and you get cleavage of the black RNA, but you also get cleavage of these green RNAs. And so we thought, when we saw this, this would not be great to put in a cell, because it probably would activate and chew up many transcripts and probably turn off genes that you may not want turned off, and maybe potentially even kill the cell. We found it actually could stall bacterial cells and kill bacterial cells. So we thought this might not be good for RNA therapeutics, but what we did realize is the function of cleaving something that's fluorescent in the presence of a specific RNA could be useful for diagnostics. And so we thought, imagine there was this blue RNA that you want to find, like for example, from a COVID-19, and you design guide RNAs that detect this RNA, and Cas13 would bind and activate when those were present, and then cleave reporters that, when cleaved, release fluorescence by separating a fluorophore from a quencher. And so we spent a lot of time developing diagnostic assays like this, and we actually made them highly sensitive in an assay called SHERLOCK, where we could couple a preamplification of other nucleic acid up front. And so in this format, we basically can take double-stranded DNA or RNA and actually amplify it using isothermal amplifications like RPA or LAMP or SDA and so on. And the amplified DNA can then be turned into RNA, which can then be detected by Cas13, resulting in detection. And since then, we've actually found that a lot of other CRISPR enzymes have this function. So even Cas12s have collateral activity, so you can use Cas12s for diagnostics. And we're finding more and more. Fung recently had this TMPB paper about ancestral CRISPR systems that also have collateral. So it's a really exciting time in the space to realize that we can use CRISPR not only for therapeutics but also for diagnostics. And so over the past few years, we've continued to develop very specific applications of these diagnostics. And so there's a lot of different areas in healthcare that could benefit. So we've shown detection of viruses, bacteria, point of care detection on lateral flow strips, patient genotyping from saliva, even cancer detection from DNA that's in the blood via kind of point of care liquid biopsies. So we've been pretty excited about the broad utility of these approaches. And when essentially the pandemic took off, we realized that there was potential that these types of assays like SHERLOCK could be really beneficial for COVID-19 detection. So I want to tell a little bit about our work from the past year and a half, continuing to improve these chemistries and diagnostics for SARS-CoV-2 detection. So basically, early on in the pandemic, this is going back now, January 2020, essentially we were getting a lot of inbound interest from other scientists and folks who were worried about what was going on in Asia and wanted us to potentially look into using CRISPR enzymes to detect the virus. And the big problem was, there's obviously qPCR and other more established nucleic acid detection methods, but they're slow and expensive, and there's supply constraints. And so there's a big interest in sort of portable point of care technologies for doing this. Imagine you could just detect yourself at home in 30 minutes. And so, early on, we actually, through SHERLOCK Biosciences, a company we co-founded, we actually were able to get FDA authorization of a CRISPR-based lab test. So this is a test that has to occur in central laboratories. It's too complex to run at home. But, I think it showed the potential that CRISPR really could be used for COVID detection. And, this was just a matter of months. It was repurposed for lab-based testing. We thought, what could we do to make at-home or workplace testing possible? And so we actually went back to the drawing board in January 2020 and thought, well, what would it take to, take an off-the-shelf, preamplification chemistry like LAMP and actually have CRISPR enzymes work in a single, reaction rather than having the multiple steps I showed before. So we actually found thermophilic enzymes from the Cas12 family that actually could work in the LAMP master mix. And what we were able to do is basically design a workflow that was incredibly simple. The idea was that someone could swab themselves, dip that swab in a lysis buffer that would break open the viral particles. then the nucleic acid would be bound by magnetic beads, which we can then pull down via magnet. Then we replace this lysis buffer with a master mix of this is what we call stop COVID, and we just incubate at a single temperature for 30 minutes to an hour, and then we can get real-time detection by fluorescence or by a lateral flow strip that we dip in. And what was cool, what was cool about that slide, or about this workflow as shown on this slide, was that we could really simplify the workflow. So what we were able to do is essentially take a rapid extraction method, whereas other methods developed out there, including our previous protocol, would require purification that would take 45 minutes. And we could combine it with a single-step master mix, whereas all the other methods, as I mentioned before, have a fluid transfer step as shown by the red arrow. And so this simplified format really could be done in about 30 minutes, which was really great. And so we took this chemistry and actually profiled almost 400 patient samples and found that the test was actually quantitative, so our time to the threshold of detection was nicely correlated with the viral load as measured by qPCR. And so it was even really sensitive down to high CT values, which are really low virus loads in the 35 to 40 range. This gave us a sensitivity of about 93% and a specificity of 98%, which is we were pretty happy with for kind of a simple point of care test. And going forward, we've spent a lot of time now really trying to get this to work with all sorts of nasal samples and making it as robust as possible. And our vision for the future, which we are working on, is to really build a device and box around this chemistry that someone could run at home, something that looks like an Apple HomePod almost, that belongs in your kitchen or living room, but that actually would allow you to swab yourself, stick into a cartridge that then reads out on this device. And so I think it's an exciting time for diagnostics because I think the chemistries are getting really mature, and the CRISPR chemistry is just the next step in making very simplified formats that can be read out anywhere. And now that we're taking the next step of really putting this on devices, we hope that the smart home of the future has really got systems that people can really test themselves. So they wake up in the middle of the night and sick and are wondering, do I have a urinary tract infection or something, or is my fever due to just a, or is it something more serious like pneumonia or strep throat or so on. And so I do think that future is achievable, and that's what we're working on by making these chemistries simple and easy and workable in the point of care sort of format. So I think going beyond diagnostics, it's obvious, as I mentioned, that there's a lot of interest in using these enzymes in cellular formats for knocking down RNAs, editing RNAs, changing splicing and regulation of RNAs. And we've done a little bit of that with Cas13, although Cas13, of course, has some toxicity concerns due to the collateral effect. So we'll show some of our work coming up with Cas13 and also go into a new enzyme called Cas711 that was recently published that has no collateral and can do very precise RNA knockdown and modulation. So I'll pass it on to Jonathan, who will dive into a lot of this exciting work. So thanks so much.
Thanks, Omar, for covering all the different things we can do on the diagnostic side with these new RNA targeting and DNA targeting tools. But as many of you know, a lot of the power of these new CRISPR enzymes is the capacity to interrogate biology inside of living cells. And so what can we do with RNA targeting tools? Well, of course, there's a lot of different RNA in the cell. There's mRNA as well as tRNA and non-coding RNAs. So the capacity to be able to target those can actually unveil a lot of different biology. So there are many different things we could do with these tools. We've actually demonstrated a couple of things in the potential therapeutics and basic biology front, including the capacity to use their endogenous nuclease capacity for RNA knockdown, as well as the ability to do base editing. So by fusing different deaminase domains to these enzymes, we can actually be able to make specific base edits. Now, we don't have time to cover both of these today, but we'll talk a little bit about what we can do on the RNA knockdown front using both the Cas13 enzymes as well as new enzymes we've very recently discovered. Now, you could think it might be very easy to apply Cas13 inside mammalian cells, and to an extent, that's correct. So we can actually take Cas13 and put it in mammalian cells with a CRISPR RNA targeting a transcript. Here we use a ratiometric reporter that has a Gaussian luciferase and a cryptidine luciferase. By targeting one of the luciferase, here the Gaussian, we can actually measure the ratio between the two and see the extent of knockdown. And if we do this in HEC293FT cells, we can actually see that depending on how we actually fuse either cytoplasmic or nuclear localization sequences to the Cas13a, we can actually get knockdown levels as good as or better than RNAi. So you can see with both guides 1 and guides 2, with especially the nuclear Cas13a, we can get improved knockdown. And that's not a small feature, the capacity to be able to localize these enzymes to different compartments, because when targeting things like non-coding RNAs that are primarily resident in the nucleus, we can actually be able to target those better than things like RNAi. However, we want to be able to look at the specificity of these systems. So if you look at the specificity, you can take a targeting guide and a non-targeting guide and actually be able to compare using transcriptome-wide sequencing the amount of knockdown. So if you take a normal targeting guide against Gaussian luciferase and we actually plot it, here is the RNAi version, and you can see across that middle axis there's a lot of fuzziness. There's a lot of transcripts that are either upregulated or downregulated. And however, if you take the Cas13a and overlay it, you can actually see it's much more tight distribution. And that small dot off to the lower right, that's actually the Gaussian luciferase being knocked down. So it's a much more precise enzyme, at least in these cells. And this is true for multiple Cas13s. So if we do the same experiment with the Cas13b from PSP, you can see that we actually have a similar level of specificity. However, even though we did this in HEC293FT cells, it doesn't necessarily hold across all cell lines potentially. Because as you remember, these Cas13s as well as the Cas12s have this collateral activity where when they're activated, they chew up many different RNAs. So if you actually target in certain cell lines, including U87 glioblastoma cell lines, you actually see that, and this is done by a different group, but you can actually see that there's a loss of viability and reduced cell growth. And if you actually run the ribosomal RNA on a gel, you see a loss of integrity. So this really opens up a question: can we actually make new RNA targeting tools to reduce the toxic effects of these things like Collateral and provide better transcriptome perturbation methods? And of course, we want to go back to the drawing board, which for us is natural discovery. So you'll remember that Omar kind of brought up this breakdown of CRISPR enzymes coming from class 1 and class 2. And you recall that the class 2 enzymes are much more easy to use because everything is fused together. All these functions come with one single protein. However, the class 1 enzymes also often have many great functions for both DNA and RNA targeting. It's just that because they're not a single protein, they are harder to express and use in mammalian cells. However, any sort of metric or framework is always meant to be broken. So you can see that basically these class 1 systems have multiple proteins. So these type 3 enzymes, where all of these different parts are concatenated together. So in normal type 3 systems, you can see that there's multiple subunits called Cas7s, as well as sometimes a Cas11 subunit. Here, in these type 3E systems, the Cas7s and the Cas11 have actually been concatenated and fused evolutionarily into a single protein with 4 Cas7 subunits and 1 Cas11 subunit. This we call Cas711, and we actually, in a recent publication, showed that it functions as an RNA targeting enzyme, much like the other type 3 systems. However, because it's a single protein, this has two largely significant findings. One, that class 1 enzymes and class 1 systems don't always have multiple different proteins, and here this actually breaks that terminology and that evolutionary classification. Secondly, because it can actually function as a single enzyme, we thought that it might be able to be very useful as an RNA targeting system in mammalian cells. So one interesting note is that type 3 systems, while RNA targeting, lack the collateral activity of the Cas13 systems. And we can actually see that in both biochemical and E. coli-based assays. So if you do a biochemical collateral assay where we incubate with a bystander reporter RNA that's fluorescent and a trigger RNA, you can see that the only large curve coming up here is coming from the LWA Cas13A, and that the Cas711 has no activity on that collateral reporter because it has no collateral activity. Secondly, if you look for this effects of this in E. coli, when you do targeting with a LWA Cas13A, you get a reduction of growth. So if you have the RFP targeting guide there, you actually, when you're cutting in these E. coli that express RFP, you actually have a large reduction of growth. With Cas711, you see no growth effects. So here we have an enzyme that can actually function as an RNA targeting system as one protein without collateral. So the question is, how does it function when we put it into mammalian cells? So we actually used a very similar assay targeting a ratiometric luciferase reporter with a Gaussian luciferase and cryptidine luciferase. And we actually targeted those by co-expressing a Cas711 and a CRISPR RNA targeting Gaussian, and we measured luciferase. And we didn't just measure luciferase in that condition, but we also compared it to the whole family of different Cas13s as well as RNAi. And you can see that compared to many of the Cas13s, Cas711 actually has just as good or better knockdown compared to Cas13a. Now, comparing this to our findings that Cas13s don't have collateral activity in HEC293F T cells, we saw that reproduced. So if you knock down this reporter transcript, you don't see any effect on cell viability in HEC293F T cells. However, we wanted to check this against multiple different cell types. So if you then compare the knockdown efficiency in U87 cells, first we saw that Cas11 still achieves great knockdown, and RNA actually does not work in U87 cells, which is a known phenomenon. But if you look at the viability of these cells, you actually see that we get a large decrease in cell viability for the targeting conditions with many of these Cas13Bs. So you can see that in comparing the targeting and non-targeting, and often just expression of the Cas13 itself, you can have a large decrease in cell viability compared to the Cas711s, which saw a minimal decrease in cell viability and no difference between the targeting and non-targeting cell viabilities. So there you see that there's a substantial decrease in these viabilities, which is due to the large penalty of the collateral. So with Cas711, we're adding yet another protein in this arsenal that allows us to have a large range of applications. So although we've just talked about knockdown today, you can imagine that RNA binding proteins enable a huge swath of different potential applications by being able to fuse different proteins or domains to them. We can modulate translation, and we can affect exon skipping, so we can modulate splicing. We can alter localization, and we can even affect the epitranscriptome, which actually some groups have shown with Cas13. So we think that Cas13 and now Cas711 represent a growing RNA targeting CRISPR toolbox. So if you think about all the different things we can do for DNA targeting, CRISPR-I, CRISPR-A, base editing, prime editing, there's a huge amount of things we can do there on the bottom. And on the top, now in the RNA targeting toolbox, we do have a wide and growing range of applications. And these really come back to the capacity to mine natural diversity for biology and biotechnology. So we kind of want to leave people with this thought of what else is in this tree of life that will allow us to create new molecular tools. And just to give you a preview of what we're thinking of in our lab, we're thinking about, of course, tools for precise RNA modulation, and we're continuing to develop both Cas711 as well as some Cas13 tools for expanding that RNA targeting toolbox. We're also very interested in the capacity to be able to do efficient programmable gene integration without relying on endogenous repair machineries or double-stranded breaks. So being able to actually put large pieces of DNA into the genome is something that we're very excited about, both for its therapeutic as well as its basic biology applications. And once again, mining natural diversity, we think is a great way to get us there. So of course, as part of this cell engineering toolbox, we love the capacity to be able to actually measure RNA states and use those to both non-invasively measure and see where cells are without destroying them, as well as using that capacity for circuits to control these cell states. And we're very excited about using natural tools, once again, from these natural enzymes, to be able to enable that. So it's something that we're very actively working on. So with that, we'd like to thank the entire Abudayyeh-Gootenberg Lab. We're actively looking for postdocs across multiple of those pieces of the cell engineering toolbox that we described. But here we are pre-COVID enjoying some sushi. And we're based at the McGovern Institute. You can find out more at abugoot.mit.edu. And we'd also like to thank the specific contributors to those stories on a lot of the work for the Stop COVID was in collaboration with the Zhang group at the Broad Institute, and the Casl discovery was in collaboration with Eugene Koonin at NIH, and it was led by Asen Ozcan. But we'd like to thank all the members of the lab who made this possible. And with that, we'd like to open up to any questions people might have.